Now let's look at the liver. We'll begin by looking at the gross anatomy of the liver. It is one of your largest organs, located within the upper right quadrant, up against the inferior portion of the diaphragm. It consists of 4 lobes: the right lobe, the left lobe, caudate lobe, and the quadrate lobe. Along the posterior surface of the liver is a sac called the gallbladder. The gallbladder stores bile. In addition, there is a falciform ligament, which is mesentery, and a round ligament, which is the remnant of the fetal umbilical vein. This is a posterior inferior view of the liver. The hepatic portal vein delivers blood from the digestive organs. The hepatic artery delivers blood from the aorta, rich in oxygen and nutrients. Blood exiting the liver will enter into the inferior vena cava vein, which delivers the blood to the right atrium of the heart. Recall that the gallbladder is situated on the posterior surface of the liver. There are several ducts associated with the gallbladder. The liver produces the bile. The bile enters the right and left hepatic duct. The bile then flows into the common hepatic duct, the cystic duct, and then into the gallbladder. It is stored in the gallbladder until fatty foods enter the duodenum, at which time it will contract. The bile then will go back through the cystic duct into the common bile duct and empty into the duodenum. Now let's look at the microscopic anatomy of the liver. The liver consists of repeating units called lobules. The cells within the lobules carry out the work of the liver, and so these lobules are referred to as the functional units of the liver. Each lobule is 6-sided, and they are about the size of a sesame seed. In this micrograph, I have highlighted a lobule. In the center of each lobule is a central vein. Blood in the central vein flows into the hepatic vein and then into the inferior vena cava vein, which returns the blood back to the right atrium of the heart. At each of the 6 corners of the lobule is a structure called a portal triad. Triad refers to 3. There are 3 tubes that make up this structure. The bile duct that receives bile synthesized by the liver cells, a branch of the hepatic portal vein receiving blood from the digestive organs, and a branch of the hepatic artery which receives blood from the aorta. Liver cells are called hepatocytes, and they are arranged in plates called hepatic cords. Between the plates, or the hepatic cords, are fenestrated vessels called hepatic sinusoids. Located within the blood in the hepatic sinusoids are stellate macrophages. Remember, these are phagocytic cells. The hepatocytes synthesize the bile, and the bile then goes into tiny ducts called bile canaliculi, which will then drain into the bile ducts. This slide has listed all of the structures that I mentioned in the previous slide. This illustration demonstrates the sinusoids in between the hepatic cords. The liver is an amazing organ. It has a high regenerative ability. In addition, the hepatocytes carry out over 200 functions. We're just going to look at a few. The first function is the synthesis of bile by the hepatocytes. Bile consists of bile salts, which are derived from cholesterol, bile pigment, which comes from bilirubin, the breakdown product of hemoglobin, Cholesterol. This is your way of eliminating cholesterol from your body. Phospholipids, such as lecithin, and electrolytes. Remember, the biosalts are involved in emulsification of triglycerides. Biosalts and lecithin are components of the micelles, which are used to ferry the hydrophobic substances down to the absorptive cells in the small intestine. What stimulates the release of bile from the gallbladder? The hormone CCK, which is released when fatty foods enters the duodenum, causes the gallbladder to contract, releasing the bile into the duodenum. A second function of the hepatocytes is glucose metabolism. Glucose can be synthesized from non-carbohydrates, such as proteins and fats, and that process is called gluconeogenesis. In addition, hepatocytes can take glycogen that is stored in the liver and break it down into glucose through the process of glycogenolysis. Detoxification of harmful substances would be a third function. The organelle peroxisomes is important in this function. In addition, fat-soluble substances, such as medications, can be converted into water-soluble substances that can be eliminated from the body through the kidneys. Most of your plasma proteins are synthesized by the liver, and this is the fourth function. Included in these plasma proteins are albumins that are important in regulating the osmotic pressure, as well as some clotting factors. Fat-soluble vitamins are also stored in the liver, and this is your fifth function. The last and sixth function that I have listed here is the synthesis of thrombopoietin. Recall, thrombopoietin is a hormone that goes to the red bone marrow and stimulates the production of platelets through the process thrombopoiesis.